Today in class, we are going to learn how to use a microscope. Can everyone please go up and collect the microscope from the cabinet? Stop! What are you doing? That's not the correct way to hold a microscope. You have to hold it by the arm and the base.
What's the arm and the base?
This is the arm, and this is the base. You have to have one hand on the arm and one hand on the base to properly support the microscope. Now go try again. First, we need to learn about the parts of the microscope. The first step is to plug in the microscope. To turn on the light of the microscope. You can control the light intensity with the rheostat light intensity control. Located under the stage is the diaphragm. It controls the amount of light allowed through to the slide. Everyone take the slide given and place it on the stage and secure it in place with the stage clips. This is the course adjustment knob and this is the fine adjustment knob. The course adjustment knob should always be used on the lowest objective. It changes the focus in a great manner. The fine adjustment knob changes the focus in a minute manner. On this microscope, there are 4 objective lenses. The lowest power is the 4x objective lens, which is always used first. You can switch to the 10x, the 40x, or the 100x objective lens to magnify what you see on the slide. Now we are going to put what we just learned into use. We're going to be looking at a very rare species under the microscope, the basic white girl. Now that we have learned how to adjust the brightness and focus, we need to put our own slide into focus. Great, now our slide is in focus, but it is still not right. The specimen is too dark to view it clearly. Use the rheostat light intensity control to adjust the intensity of the light. Alright, let me check again. Whoa, that's too bright. Try again. Much better. Now our specimen is in perfect viewing condition. Please go to page 96 and answer the following questions about the basic white girl.
I have a question. What's a confocal microscope?
Confocal microscopes are slightly different from the compound one we use in class. Confocal microscopes work with the help of laser light. This light passes through a pinhole opening and hits the dichromatic mirror. The mirror reflects the light to the objective lens. This leads the light to the focal plane where the specimen is found. Some laser light is passed back through the objective lens and the dichromatic mirror up to a second pinhole found near the photomultiplier detector at the top of the microscope. On the board are pictures of plant tissue and stem cells being viewed under a confocal microscope. The reason these specimens are being viewed through a confocal microscope rather than a compound microscope is because of the depth of the specimen. Confocal microscopes allow us to view specimens as thick as 100 micrometers and at a higher resolution than we would be able to with a compound microscope.